Today we discuss about the topic blotting techniques. In this chapter we discuss about definition, introduction, southern blotting, northern blotting and western blotting. First, definition of blotting techniques. The blots are techniques for transferring DNA, RNA and protein onto a carrier so they can be separated and often follows the use of gel electrophoresis. Blotting techniques involves the separation and transfer of DNA, RNA and proteins onto a blotting membrane. The target DNA is then introduced to a molecule in order to aid detection. And southern blotting is used to evaluate for specific DNA sequence and may be used in identification of genetic mutations and in forensics. Northern blotting focuses on RNA sequence and is helpful in accessing gene expression. Southern blotting. This technique was developed by E.M. Southern in 1975. This blot is used to detect the presence of particular piece of DNA in a sample. The DNA detected can be single gene or it can be part of larger piece of DNA such as viral genome. Next, this blotting combines agrogel electrophoresis for size separation of DNA with methods to transfer the size separated DNA to a filter membrane for probe hybridization. The key to this method is hybridization. Hybridization means the process of forming a double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target patient DNA. In southern blotting, the mixture of molecule is separated, the molecules are immobilized on a matrix, the probe is added to the matrix to bind to the molecules and any unbound probes are then removed. The place where the probe is connected corresponds to the location of the immobilized target molecule. This is a southern blotting techniques method. In this method, the digested DNA with an appropriate restriction enzyme. The complex mixture of fragment is subjected to gel electrophoresis to separate the fragment according to size. The restriction fragment present in the gel are denatured with the alkali and transferred onto a nitrocellular filter or nylon membrane by blotting. This procedure preserves the distribution of the fragments in the gel, creating a replica of the gel on the filter. The filter is inhibited under the hybridization condition with a specific radio-labeled DNA probe. The probe hybridizes to the complementary DNA restriction fragment. The excess probe is washed away and the probe bound to the filter is detected by autoradiography, which reveals the DNA fragment to which the probe hybridized. In southern blotting techniques, extract and purify DNA from cells. DNA is restricted with enzyme, sort by electrophoresis, denatured DNA, transferred to nitrocellular paper, blocked with excess DNA, wash of unbound probe and autoradiograph. Northern blotting technique is for detection of specific RNA sequence. Northern blotting was developed by James Alvin and George Stark at Stanford University in 1979 and was named such by analogy to southern blotting. It is isolated from several biological samples. Example, various tissues, various developmental stages and same tissue, etc. RNA is more susceptible to degradation than DNA. The gel is then blotted onto a nylon membrane or a nitrocellular filter paper, diazophenoxylene methyl papers by creating the sandwich arrangement. This arrangement is called sandwich arrangements. The samples are loaded on gel and the RNA samples are separated according to their size on an agrogel. The resulting gel following after the electrophoresis strand. The membrane is placed in a dish containing hybridization buffer with a labeled probe. Thus, it will hybridize to the RNA on the blot that correspond to the sequence of interest. The membrane is washed to remove unbound probe. The labeled probe is detected by autoradiography or by chemiluminescence reactions. In both cases result in the formation of dark band on X-ray film. Now the expression pattern of the sequence of interest in the different samples can be completed. Next, the standard of the uses of northern blotting is to standard for the study of gene expression at the level of mRNA, detection of mRNA transcript size and study of RNA degradation, RNA splicing and RNA half-life. Often used to confirm and check transgenic or knockout mice, examples animals. In Northern blotting techniques, the standard Northern blot method is relatively less sensitive. The detection with multiple probes is a problem. If RNA sample are even slightly degraded by RNA, the quality of data on quantification of expression is quite negatively affected. Next, we will see about Western blotting. Western blotting 1981 is an immunoblotting technique which rely on the specificity of binding between a protein of interest and a probe to allow detection of the protein of interest in the mixture of many other similar molecules. This page technique is prerequisite for Western blotting. In Western blotting, protein sample is subject to electrophoresis on an SDS polyacrylamide gel. Electron blotting transfers the separated proteins from the gel to the surface of nitrocellulose membrane. The blot is inhibited with the generic protein such as milk protein or BSA, which binds to any remaining sticky place on nitrocellulose. An antibody that is specific for the protein of interest is added to the nitrocellulose sheet and react with the antigen. On the band containing the protein of interest binds the antibody, forming a layer of antibody molecules. After washing for removal of non-specifically bound antibody 1, second antibody AB2 is added, which specifically recognizes the Fc domain of the primary antibody and binds it. AB2 is radioactively labeled or as covalently linked to a reporter enzyme, which allow to visualize the protein AB1 AB2 complex. Thank you.